Hey everyone, welcome back to another episode of Genetics. Today we're going to continue in Chapter 7 talking about eukaryotic transcription. We've already talked about prokaryotic transcription and how to initiate, and we've also talked about the different types of mRNA that you can find. But today we're mainly interested in eukaryotic transcription, and then next episode we're going to go over the different types of eukaryotic modification that they do to develop a mature mRNA molecule. Okay? So to begin with, we're going to talk about transcription initiation complex, and then we're going to talk about two different sequences, enhancers and silencers, and what they do to initiate transcription. Okay? So the first step is basically to initiate transcription in eukaryotic organisms. What you need to do is you need to find a promoter sequence, and typically these promoter sequences on these genes have multiple different types of regulatory sequences, like something like a Tata box, and there could be many other different types of regulatory sequences in a promoter sequence. So these transcription these initiation proteins will bind to each other, and they'll bind to that correct box, to the Tata box, for example, and they'll begin to make a initiation complex out of multiple different types of proteins. So here's an image from your textbook. It basically goes over the different kinds of proteins that will be combined together to make this complex. To be honest, you probably shouldn't memorize what these are. All you have to do is memorize that there's a huge complex being made for transcription in eukaryotic organisms. As soon as that complex is also formed, what you're going to add, you're going to add a RNA polymerase. Now, interestingly, eukaryotic organisms have multiple different kinds of RNA polymerases. As you can see here in the image, they specify RNA polymerase 2. But you can have different RNA polymerases, and they just all have a different function that they're trying to synthesize. So I believe RNA polymerase 1 was the one that was responsible for tRNA. So on and so forth. Every single RNA polymerase has a different goal that they're trying to synthesize. One is trying to synthesize mRNA. The others are trying to synthesize some sort of other functional piece of RNA. So basically, so like I said, this initiation complex forms, and then when it forms, you begin transcription, and then these initiation factors pop off, they go away from one another, and that's essentially how you end up with transcription in eukaryotic organisms. Okay, so pretty simple. And then when we go into the next thing that I wanted to go into is enhancer and silencer sequences. So what these sequences are found really far upstream or downstream of the promoter region. And basically what these sequences do is a protein will bind to an enhancer sequence or a silencer sequence, and then when that protein binds to that enhancer or silencer sequence, it will form a tighter association with the RNA polymerase. And if it's an enhancer sequence, what will happen is you would promote the transcription of the gene. But if you have a silencer sequence and a protein is bound to that silencer sequence, what will end up happening is you would repress the transcription of the gene. So essentially, this is how that whole entire complex looks like, where you have different proteins that bind to the enhancer sequence, and when they bind to the enhancer sequence, they also promote the pull of RNA polymerase 2 to the initiation complex, and as a result, you have more transcription than if you would have it before. But that's basically it for today's episode. It was pretty short. To be honest, you don't really need to know all the different proteins associated with the initiation complex. There's way too many. But just understand that it's a lot more complicated in eukaryotic organisms, and there's different sequences that are contributing whether you're going to have transcription of a gene and whether you're not going to have transcription of a gene. But yeah, that's it.